Hi there. In this short video, we're going to talk about program management on Pluto, and then we'll go into a rare disease example where we identify and manage biomarkers from the data. So what you're seeing here is the programs view in Pluto where you can track and manage all of your programs in one place. We can convert this to a Kanban style view and then narrow this down to biomarker identification programs. And then let's go into this progeria rare disease example. So here you can see a summary of the program. And then importantly, you'll see the translational alignment framework that shows all of the different analyses and steps that are recommended to complete a really robust, well-supported program. You can also see the declared target of the program, intent and scope, and importantly, the timeline, which is mapped here. When we're really ready to start analyzing data, we can go into data sets. And then you'll see here a data set that is RNA-seq taken from mice, and this is actually a public data set that was pulled in just by using the GeoImports tool on Pluto. So when we navigate into that data set, we can see a high-level overview of things like samples and metadata variables, and then see a breakdown of the samples based on all of those different attributes. And then you'll also be able to see the assay data and the sample metadata. Analysis on Pluto can start with either FASTQ files or counts data. This is an example of one that just starts with counts data. But once that counts data is ready to go, then we can actually begin analysis. So we can head to analysis space, and you'll be able to see all of the recent analyses that were run, any evidence and targets or biomarkers that were pulled from that analysis. And then you can also see a report on the analysis. So information like the scientific question, the study design, any recommended analyses, for example. To actually start generating visualizations, you can head to the canvas. So at the top, I've just defined our question here. Do specific BUBR1 allelic combinations produce distinct transcriptional programs that explain phenotypic heterogeneity in MVA progeria? And you can also see the hypothesis and goal at the top here. So all of these analyses that you see here are run using graphical interface modules. So for example, if I click into this differential expression analysis, the analysis was just run by selecting experimental groups, and those groups are pulled from the metadata that is uploaded by the user. And you can also create combinations of variables. For example, here I've combined condition and tissue to be able to select this HH genotype gastroc muscle versus wild type gastroc muscle, and then just run that analysis. And then the user can change the plot however they want. So they could change it to be a heat map, they could change the p-value or the log fold threshold lines, and they can change the colors. And then importantly, you have the option to label entire lists of biomarkers or to select individual genes from the target list. And you can create lists of targets anew. So let's say that I wanted to just lasso all of the upregulated genes in the HH genotype condition. I could lasso that selection and then save those genes to a new list. So we could call this MVA progeria HH genes up. Save that as a biomarker list. And then if you go to biomarker management, you can select that list and see all of the biomarkers that were just saved. And you can also store evidence like visualizations and notes here as well. You may want to save a specific plot as evidence of a certain biomarker, in which case you can click on that visualization and save as evidence. And then we can select a biomarker list and select that biomarker. So let's say I want to note RPL29, and then we can go to evidence in this analysis space, and we'll see that that biomarker has been saved. We can view the details and see a report on that biomarker, as well as an AI summary of the gene, and then you can see the evidence that was just saved related to that biomarker. So hopefully this gives you an idea of not only how analysis is run on Pluto, but how you can link the data that comes out of that analysis to biomarker discovery and manage that all in one place.